All right. I would like to welcome Amelia Stout, who's going to present temporal surveillance of Coccidioides and aerosols in soils at a single location in Arizona.
Hi, everyone. Thank you for being here. I am excited to get the opportunity to talk with you. So as I think all of us here know, valley fever is an environmentally acquired disease. It's a soil-dwelling fungus, but the primary infection mechanism for humans is inhalation of the fungus through aerosol dust. So today I'm going to be talking about aerosol surveillance in Arizona. In highly endemic regions, such as Arizona, up to 50% of long-term residents have been infected with coccidioides, and aerosolization and inhalation of the fungus is the key pathway to infection. To date, there have been no temporal studies that examine the presence of coccidioides in the soil and air at a single location within the hyperendemic region for coccidioidomycosis and relate that presence to soil and air variables. A few studies have shown the presence of coccies in air, but focus on a few points in time. The goal of our research was to analyze the association between the presence of cocci in climactic factors as well as soil and air chemistry in assessing the temporal variability of cocci in the soil and air. We also wanted to determine if the genotypes we're seeing in the air match the genotypes we're seeing in the soil, or if there's some sort of transport happening and they're coming from more distant sources. So we have been sampling at a site in Mesa. Sampling is ongoing, but we started sampling in 2022. So today I'll be talking about our first year of sampling from September 2022 to September 2023. We do high volume PM10 sampling, and we do PM10 sampling because that's the particle size that we can actually inhale. Those are the ones that affect us. So we followed the EPA 1 and 6 day sampling schedule. So we run our samplers every 6 days for 24 hours. And we sample on 2 different types of filter media for different analyses. So we sample on quartz fiber filters, and we do ion chromatography, where we identify key ions. We do carbon analysis, where we look at elemental and organic carbon present. And we do gravimetric analysis to get a time-integrated PM10 concentration. We send half of each quartz fiber filter to NAU for biological analysis, where they do qPCR to determine presence of cocci on our filters. For cellulose filters, we do a complete HF digestion, followed by inductively coupled plasma mass spectrometry for a full elemental analysis. We also looked for cocci present in 14 different animal burrow systems and soil from 2 random locations, but for the purpose of today's presentation, I will primarily be focusing on the air data. So the figure on the left here is a timeline of PM10 over that first year of sampling. And you can see the date that's highlighted, we caught a haboob. So there's higher PM10 than any of our other sampling days. And the dates that are marked with a star are dates that cocci were detected on our filters. So we are sampling at a known hotspot, but even so, we were still surprised to see such high rates of positivity. Every sample from the middle of January to the middle of September was positive, so definitely a unique site in terms of positivity. On the right, taking a closer look at the relationship between PM10 concentrations and positivity, we see that higher PM10 concentrations are related with higher presence of cocci in the air. Our colleagues at NAU have developed a model to determine probability of occurrence of cocci. It's a Bayesian multi-scale occupancy model with a hierarchy of parameters. So the first parameter specifies the probability of the species at the burrow. The second parameter is the conditional probability of the species of occurrence within the soil sample. And the third parameter is the conditional probability of the species detection within each PCR replicate. So taking a look at the output of that model, on the left, we have the probability of occurrence in the air, and on the right, we have the probability of occurrence in the soil. And we see that the soil has distinct patterns of probability positivity that are not observed in the air, and the air has much greater temporal variability. This really emphasizes the importance of entrainment, because we see that it's almost always present in the soil, but it's not always present in the air. This leads us to wonder if there's possible transport and if the cocci are coming from more distant sources, which leads us to look at meteorological and chemical parameters to give us an indication of the source. So first we looked at wind speed, and we saw that both average wind speed and maximum wind speed have a strong relationship, with higher wind speeds being associated with higher presence of cocci in the air. Which makes sense because at those high wind speeds, we typically see more dust entrainment and more particulates getting in the air. We didn't see a relationship with wind direction or other meteorological parameters. So enrichment factors tell us how enriched a sample is compared to a reference. So here we're looking at the enrichment of the aerosols compared to a reference of upper continental crust. And we see that most elements are around 1, which tells us that they're not enriched and that they are reflective of the reference that we use. So it's reflective of the soil contribution. We do see a few elements are enriched. So, and I do want to point out this is a log scale. So we see that copper, molybdenum, and antimony are all enriched. But that doesn't necessarily indicate anthropogenic activities or impacts. So taking a closer look at a couple of those elements, copper and antimony, we see that they're enriched, but they're consistently enriched throughout the year. We see a couple of elevated points on December 31st, so there may have been more anthropogenic activities happening then, such as fireworks. But throughout the year, they're consistently elevated. So this is likely not due to anthropogenic activities, but more likely that it's a unique composition of the local soils that differ from the reference of upper continental crust that we looked at. Here we're looking at a reconstruction of the PM10. So there's crustal material, carbon, which is made up of organic carbon and elemental carbon, and then ions. And we see that the crustal material represents a significant amount of the measured PM10, and on average represents over 40%. When comparing that to a more urban location, such as Phoenix, we see it significantly more. Data from Phoenix, it was collected from the Improve Network from an EPA monitoring site located in central Phoenix. And we see that both sites have similar amounts of PM10. Mesa has 19.6 micrograms per cubic meter, and Phoenix has 20.9 microgram per cubic meter. So they're very similar in concentrations, but the sources are different. So we see that Mesa has much more crustal material, which tells us that it's more dependent on soil entrainment. So we saw that, or we know that entrainment of cocci is the essential step for infection of valley fever in humans. Both at the Mesa site, both the soil and the air are highly positive for cocci, and there's low temporal variability of the aerosols when looking at chemical parameters. We saw strong relationships with higher PM10 and higher wind speeds being related to higher presence of cocci in the air. But the most important thing that we found was that the occurrence of cocci in the air does not match the probability of occurrence in the soil, which again really emphasizes the importance of entrainment. We have a lot of work ongoing, so further efforts to investigate remote versus local sources of the crustal material by looking at molecular markers such as carbohydrates, looking at rare earth elements to give us a more detailed fingerprint of the aerosols and the soils at the site to see if they're coming from the same, or to see if the aerosols are coming from the soils at the site or if they're coming from somewhere else. And our colleagues at NAU have been doing a lot of work on genotyping. Dan and Nate have a really interesting poster. I think it's poster 56. So go check that out, and they link genotypes that were found on the air filters to known infections and genotypes found in the soil. We're also working on a second hotspot in southern Arizona, and sampling at the Mesa site is ongoing, and the data series will soon be extended. I'd like to thank Arizona Board of Regents for funding this project and our collaborators at NAU for all of their work. Thank you. I'd be happy to take any questions.
Okay. Okay, questions?
Yeah, looking at the miles per hour of your wind speeds, I guess I'd like to emphasize it's not super high Habib-like wind speeds. I mean,
Yeah.
So it's a statistical effect, but I think it speaks towards the risk of inhaling a cocci spore is kind of an ambient risk across the 24/7, more so when it's a little windier, less so when it's less windy, but it's not really dependent on those photogenic haboobs that we get in the summertime.
Thank you for sharing your data. When you speak about a distant transport, what sort of distances are you considering? Is it meters, miles, hundreds of miles that you think might be important?
I would probably guess miles. I think, I don't know if hundreds of miles. I mean, maybe it could transport that far, but I think miles would be more likely.
And I was also curious, in that case, would sampling the air at a nearby site, but not right in the hot spot, also help inform long distance transport on the order of miles to help? And are you doing that? Are you sampling at some like nearby, but not?
No, right now we're only sampling at this location. I think that could give us a better indication, especially if we were able to genotype everything that we're finding, seeing if we're seeing the same things in different locations. But right now we're just sampling at this one site in Mesa and then a site in southern Arizona.
Thanks.
Can you go into more detail about trying to match up the geolocation using heavy metals and some rare earth minerals, I think you said, and some other aspects?
Yeah.
Because we've tried to do that with wildfire smoke to see where the geographic range it started, and it's actually very difficult to do. And if this is just a few miles apart, I'm just curious how you all have approached that. With that slide, yeah.
Well, I think at the very least, we would be able to tell that the aerosols are coming from somewhere other than our site. So I think that would be the first step, would be determining that the aerosols are coming from somewhere else, seeing that they don't match the local soils necessarily.
So you're looking at total soil composition, not just because I thought that said
Yeah,
and
it would be
coccidioides.
looking at rare earth elements. So
Out of the spore, out of cocci though?
no,
Okay.
out of the particulate matter. So
Okay.
we're looking at the PM10.
That's simpler. All right.
Yeah.
Okay, thanks.
Yeah.
More questions? If not, thank you.